Imagine standing in your kitchen, pouring a handful of quinoa into a pot of boiling water. Within minutes, the tiny seeds begin to swell, and a delicate white ring detaches from the core, curling around it like the halo of a miniature planet. We casually call it a grain. We place it on the same shelf as rice, wheat, and barley. Yet biologically, this is a centuries-old misunderstanding. Quinoa does not belong to the grass family. It is not related to wheat. It is, in fact, a cousin of spinach and beets. A perfect impostor, not because it intends to deceive us, but because its evolutionary design is remarkably sophisticated. Inside each seed lies a survival blueprint, written in chemistry, sunlight, and the thin atmospheric pressure of the Andes Mountains. In botany, a cereal refers specifically to the seeds of plants in the Poaceae family. The grasses, wheat, rice, and corn share a defining biological structure known as a caryopsis, in which the seed coat is fused to the fruit wall. Quinoa is different. It belongs to the species Chenopodium quinoa, part of the Amaranthaceae family, the amaranths. The term pseudo-cereal was coined to describe plants that are not grasses but are used like grains because of their starch content and similar preparation methods. But quinoa's identity crisis is not merely a matter of classification. It lies in the anatomical architecture of the seed itself. When you cook quinoa and observe the white halo unfurling, you are not witnessing a random rupture of the seed coat. That white ring is the embryo, the germ coiled around the endosperm like a galaxy embracing its central planet. In wheat, the embryo occupies only a small portion at the base of the kernel. In quinoa, the embryo wraps around nearly the entire seed. Imagine this structure as a two-layered vault. At the center sits the starchy core, an energy reserve. Encircling it is the embryo ring, a biological command center containing lipids, enzymes, and proteins. This configuration allows quinoa to activate rapidly during germination, because the growth engine is positioned directly around its fuel supply. If you have ever forgotten to rinse quinoa and noticed an unpleasant bitterness, you have encountered its chemical armor. The compound responsible for that taste is saponin, a group of glycosides composed of a sugar component, glycone, attached to a triterpenoid, or steroid aglycone. At the molecular level, saponins function as natural surfactants. They can interact with the cell membranes of insects and small animals, disrupting the integrity of the lipid bilayer. Picture a cell membrane as a double-layered lipid fortress wall. Saponin acts like a chemical key capable of inserting itself into that wall and destabilizing its structure. The result: birds and insects avoid untreated quinoa seeds. Without synthetic pesticides, the plant has equipped itself with a biochemical defense system. Notably, saponins are concentrated primarily in the outer seed coat. When humans developed washing and abrasion techniques to remove them, we were essentially stripping away the armor nature designed for survival. Here lies an intriguing paradox. What we perceive as a flaw in taste is, in reality, a protective adaptation. And in modern agriculture, as many quinoa varieties are bred to contain lower levels of saponin, we may be unintentionally weakening this natural defense, trading resilience for convenience and a milder flavor. In nutrition science, the concept of a complete protein is typically associated with animal-based foods, because they contain all 9 essential amino acids that the human body cannot synthesize on its own. In plants, this is relatively rare due to imbalances in amino acids such as lysine, methionine, or tryptophan. Quinoa is the exception. Biochemical analysis shows that it contains all 9 essential amino acids, with significantly higher levels of lysine than wheat or corn. Lysine plays a crucial role in collagen synthesis and calcium absorption. Methionine participates in methylation cycles and detoxification processes. Tryptophan serves as a precursor to serotonin. Think of the amino acid profile as a biological alphabet. To write the story of proteins in the human body, you need the full set of letters. Missing even one letter interrupts the manuscript. Quinoa provides the complete character set in one compact package. The protein structure of quinoa also contains globulins and albumins, forms of protein that are more easily digested than the gluten found in wheat. This is why quinoa is naturally gluten-free and suitable for individuals with celiac disease. It is not by chance that quinoa is considered a biological machine. Its molecular design reflects an evolutionary strategy storing all necessary components so that the embryo can initiate life even in harsh environments. Humans, incidentally, benefit from that design. Quinoa did not originate in fertile plains. It evolved on the Andean highlands, at altitudes reaching 4,000 meters. There, atmospheric pressure is low, reducing oxygen availability. Ultraviolet radiation is more intense, and the temperature difference between day and night can span dozens of degrees. To survive, quinoa developed multiple adaptive mechanisms. Its leaves can regulate stomatal openings to minimize water loss. Some varieties accumulate betalains, powerful antioxidant pigments that help protect cells from UV-induced damage. Its root system can extract nutrients from poor soils and even tolerate high salinity. At the physiological level, quinoa's photosynthetic process exhibits flexibility under environmental stress. It can maintain relatively stable efficiency even when temperatures fluctuate dramatically, something many staple crops cannot achieve. Because of these characteristics, quinoa has been examined in research on closed agricultural systems, including potential applications in space missions. When humans consider cultivating plants in resource-limited environments, they turn to species that nature has already trained under extreme conditions. Quinoa is not merely a crop. It is the result of thousands of years of natural selection in environments where many other species failed. We call quinoa a superfood as a fashionable label. But what makes it exceptional is not trendiness. It is evolutionary logic compressed into a small seed, an embryo structure surrounding a starch core like an intelligent design, a bitter layer of saponins acting as chemical armor, a complete amino acid profile resembling an optimized genetic code, and resilience to extreme environments as a declaration of survival. Quinoa is not an impostor in any negative sense. It is proof that human classifications are sometimes too simple in the face of nature's creativity. In the botanical world, it is not a true cereal grain. But in the world of evolution, it is a complete answer to the question, how does life continue when every condition seems to oppose it?